Assalamu alaikum. This video is a continuation of the previous topic on the T cells, and in this video we are going to talk about the activation of the T cells. In our lecture of inflammation, we talked about that when is there an injury, the organisms or pathogens, they get a portal of entry through which they enter into the body, and the body produces an inflammatory response against them. As a result of that inflammatory response, phagocytes, which include both macrophages and neutrophils, they appear at the site of infection. They engulf those pathogens, and their lysosomal enzymes disintegrate those pathogens. And now here on, there are two different types of mechanisms. The macrophages, they present those antigens that are a part of those bacteria. They present those antigens on their surface in association with the class 2 MHC protein. But neutrophils, they disintegrate the bacteria and they excrete the remains of bacteria in the lymphatics. We will talk about it later in this video. First we will focus on the macrophages. So the macrophages present the antigen on their surface along with the MHC protein, which is of class 2. We talked about the MHC protein, that there are three types of MHC proteins, but two, class 1 and class 2. Both of these are important in the immune response. Class 1 MHC protein is for the endogenously synthesized antigens, which include viral proteins, and the class 2 MHC protein is for the extracellular microorganisms. So the macrophage, which is also called as the antigen presenting cell, it will take that antigen in association with the MHC protein on its surface, and it will move towards the T cells, helper T cells, which are also called as CD4 positive T cells. These macrophages will present that antigen to the T cells. T cells have CD4 on their surface, and that CD4 molecule will recognize the type of MHC protein which is present on the surface of the macrophages or antigen presenting cells. The TCR, which included CD3, which is a complex of CD3 and antigen receptor, it will recognize the antigen on the surface of the APCs or antigen presenting cells. This is the first interaction that is taking place between the macrophage and the T cell. There is also a second interaction between these cells. There is a protein present on the T cells which is called LFA1 protein, and this LFA1 protein will bind with the ICAM1 protein which is present on the surface of the antigen presenting cells. So the LFA1 and ICAM1 will interact with each other, and this is the second interaction between the macrophages or the antigen presenting cells and the T cells. That there is also a third interaction that is taking place between these two cells. The T cell also have another molecule on its surface which is designated as CD28, and this CD28 recognizes a protein molecule that is present on the surface of the antigen presenting cell or macrophage, which is called as the B7 molecule. Now the B7 molecule will interact with the CD28, and now after this the T cell is fully activated. The T cell, which is the helper T cell or the CD4 positive T cell, and its other type CD8 which is called the cytotoxic T cells, both of these cells cannot recognize antigens if the antigens are not presented with the MHC protein, and this phenomenon is known as the MHC restriction. If the antigen is not presented with the MHC protein, then the CD4 and CD8 cells cannot recognize that antigen. Now let's talk about the neutrophils. The neutrophils, they engulfed the pathogens, they disintegrated those pathogens, and they drained their remains into the lymphatics. Through the lymphatics, some of the antigens, they reach to the B cells. B cells have receptors on their surface which are called as the B cell receptors, which are actually immunoglobulins, and these immunoglobulins include IgM and IgD. So the antigens are attached to those immunoglobulins, and the B cell recognizes those antigens as foreign and engulf those antigens. Then the B cell produces MHC protein, MHC class 2 protein, and it presents those antigens on its surface along with the MHC class 2 protein. Now the B cell is also presenting the antigens, and it will take those antigens towards the B cells and it will activate the T cells. Now when the antigen present on the APCs along with the MHC interacts with the T cell is activated. And in our previous video we talked about the CD3 molecule that it is involved in a signal transduction pathway. When the T cell is activated, a signal is transmitted, and a series of enzymes called as phosphokinases is activated, and these phosphokinases will then further activate an enzyme called as phospholipase C. This phospholipase C will cleave phosphoinositide, which is a membrane lipid, and the cleavage of the phosphoinositide produces inositol triphosphate, and this inositol triphosphate which will open the calcium channels, and this calcium then activates another enzyme which is called as calcineurin, and it is a serine phosphatase. This calcineurin moves to the nucleus and it is involved in the activation of the interleukin 2 and interleukin 2 receptor. And we talked about that interleukin 2 is a T cell growth factor. The interleukin 2 receptor will appear on the surface of the T cell, and the interleukin 2 will bind to the interleukin 2 receptor, and it will cause the further growth of the T cells. The T cells that are growing, they will perform their effector and regulatory function, and some of the T cells will become the memory cells, which are capable of being rapidly activated upon exposure to antigen at a later time. The cytotoxic T cells and B cells can also form the memory cells. We also talked about the interleukin 2, that interleukin 2 also stimulates or activates the CD8 positive T cells, which are called the cytotoxic T cells, as well as it will also activate the CD4 cells. The activated CD4 cells will produce another cytokine which is called as gamma interferon, and this gamma interferon will activate the macrophages, and it will indirectly increase the production of the MHC protein, and then the MHC protein which will be associated with the antigen on the surface of the macrophages and antigen presenting cells.